All right, today we are diving deep into one of the biggest, most exciting breakthroughs in modern medicine, chimeric antigen receptors, or CARs for short. And this isn't about some new pill, no. This is about engineering a living drug. We're talking about reprogramming our very own immune cells and turning them into these elite cancer-hunting machines.
We hear the term CAR T therapy thrown around a lot, right? But what does that actually mean? How do scientists take a normal immune cell and transform it into a precision-guided weapon? That's what we're going to unpack. And hey, if you enjoy these deep dives, it would be amazing if you could subscribe, hit that like button, and share this with your friends. It really helps us out.
So first things first, let's get to the heart of it. What is this machine we're building? The name chimeric antigen receptor sounds super complicated, but the idea behind it is actually, beautifully simple.
Okay, think of a CAR like a custom-built sensor made of 4 key parts that you can swap in and out. On the outside of the cell, you've got the extracellular domain, or ECD. That's the GPS, the part that's looking for the target. Then there's a flexible hinge, which connects it to the transmembrane domain, and that's just the anchor that holds the whole thing in place. And then, and this is crucial, on the inside of the cell you have the intracellular domain, the ICD. That's the engine. That's the part that screams attack once the target is found. And because each part is modular, well, that's where the engineering magic really begins.
So let's start with that engine, because the story of the intracellular domain, that's really the story of how CAR technology went from just a cool idea in a lab to a therapy that is literally saving lives. It's all about giving the cell the right commands to not just kill, but to survive and win what's often a very long war inside the body.
This whole evolution happened in these really distinct phases, or generations, as we call them. We started with a basic on switch. Then we figured out how to give it stamina. We even experimented with adding more power. And finally, we created CARs that can completely remodel the battlefield around them.
First-generation CARs were really the proof of concept. They had just one little signaling part, CD3 zeta, which is basically the go signal for a T cell. And you know what? They worked. They could absolutely kill cancer cells. But it was like a drag racer's engine, a huge, incredible burst of power, but they burned out almost instantly. They just didn't have the persistence, the staying power, needed for a real lasting cure.
And that brings us to this second generation, which was the absolute game changer. Scientists figured out that the T cell needed more than just a go signal. It needed a second signal, what's called a co-stimulatory signal, to tell it to survive, to multiply, and to stick around. It was like bolting a turbocharger onto that engine, giving it both raw power and endurance. But here's where it gets interesting. Not all turbochargers are built the same. Engineers found they had a really important choice to make. They could use a domain called CD28, which gives the T cell a really fast, explosive burst of activity, perfect for a quick, overwhelming attack. Or they could use 41BB, which gives a slower, more sustained signal. This one encourages the cells to last a lot longer and even form a memory response. The choice between them completely changes how the CAR T cell behaves. So the natural next thought was, hey, if one turbocharger is good, then 2 must be better, right? Well, that was the idea behind third-generation CARs. They tried to stick both CD28 and 41BB in there, but it turns out you can have too much of a good thing. Overstimulating the T cell, just pushing it too hard, can make it enter a state of exhaustion. It basically just burns out the engine, making it less effective. A huge lesson was learned here. More power isn't always the right answer.
This led to a total paradigm shift. Instead of just trying to make the CAR T cell itself more powerful, the question became, what if we could engineer it to change its entire environment? And this is the brilliant idea behind fourth-generation CARs, which have the best nickname ever, trucks.
The name says it all. T cells redirected for universal cytokine killing. So when a T-truck finds its cancer target, its engine doesn't just say kill, it also has a second command, release the payload. This payload is often a powerful immune booster, like interleukin-12, and it acts like a flare. It turns the tumor's defensive, cold environment into a hot one, basically screaming for backup and attracting even more of the patient's own immune cells to come join the fight. And this table just lays it all out perfectly. You can literally see the journey laid out, from that simple activation switch in Gen 1 to adding persistence in Gen 2. Gen 3 was this experiment in just raw power, which then led to the super elegant solution of Gen 4, not just killing the enemy, but reshaping the entire battlefield. It's just a masterclass in iterative biological design.
Okay, so we've seriously upgraded the engine, but a powerful engine is pretty useless if your GPS can't find the destination, right? So now let's shift our focus to the outside of the cell, the extracellular domain, and see how engineers are making CARs way better at finding their targets. So the classic targeting part is called an scFv, and it works. It works really well. But it's kind of bulky, and that can be a real problem when you're going after solid tumors, which build these dense fortress-like structures that are really hard to get inside. The solution? Nanobodies. These actually come from the unique antibodies found in camels and llamas, and they are way smaller, more stable, and can just wiggle their way deep into a tumor's defenses.
And the size difference here is pretty dramatic. We're talking about a nanobody being around 15 kilodaltons, which is roughly half the size of a traditional scFv. Now, this isn't some minor tweak. This is a massive structural change. This smaller, more nimble design might just be the key to finally making CAR-Ts effective against solid tumors, which, by the way, make up the vast majority of all cancer.
So now we have a better engine and a better GPS. But there's another problem. Cancer is incredibly clever. A tumor can actually escape the therapy by just stopping, just turning off the production of the very target the CAR-T is looking for. It's like a fugitive changing their appearance to dodge the cops. So how do we get around that? Well, the answer is to give our CAR-T cells more than one thing to look for. One approach is something called a TAN-CAR, or a tandem CAR. This design literally puts two different GPS systems on a single receptor. It works on OR logic. If it sees target A, it attacks. If it sees target B, it attacks. It makes it so much harder for the cancer cells to hide.
But wait, what if a perfectly healthy cell happens to have just one of those targets? An OR logic CAR might attack it by mistake. So, for more safety, for more precision, engineers developed the DualCAR system. In this setup, the T cell has two totally separate CARs on its surface. For the cell to get fully activated, it needs to see target A and target B at the same time. This AND logic is an absolutely critical safety switch, making sure these powerful cells only unleash their full force when they are 100% sure they found the right cancer cell.
Now, up to this point, we've only been talking about T cells. But the CAR itself, that's just a piece of tech. It's a guidance and activation system. And what's so revolutionary is that this system, this amazing piece of engineering, can be installed on other types of immune cells. We can literally build a whole new army.
But here is the most important part. You can't just drop a T-cell engine into a different kind of cell, into a different chassis. Each cell type speaks its own language. It has its own unique activation pathway. So to make a CAR-NK cell, or a natural killer cell, you have to swap in a signal like DAP12. That's what triggers its rapid-fire killing. For a CAR macrophage, you need a different signal, like the FcR gamma chain, to tell this big eater of the immune system to start engulfing its target. It's all about matching the hardware to the specific operating system of each cell.
Wow. Okay, so from simple on switches to these environment remodeling trucks, from single targets to complex and logic gates, and from just T cells to a whole new army of different engineered immune cells, the pace of innovation here is just absolutely breathtaking. So what's next? Well, the future is all about control and complexity. Engineers are now designing switchable CARs that can be turned on or off with a simple pill. This gives doctors real-time control to manage side effects, which is huge. And get this, artificial intelligence is now being used to design and predict how new CAR structures will perform, which is dramatically speeding up the development of the next generation of these incredible living drugs.
We've moved beyond simple drugs. We're now creating programmable, living medicines. We're not just treating diseases anymore. We are literally teaching our own bodies how to cure themselves from the inside out. The journey began with cancer, but this platform, this technology, can be applied to so much more. And that leaves us with this thrilling question to think about. As we get better and better at this, what's the next frontier? What disease will we teach ourselves to cure next?